Hi everybody, I'm Professor Patrick Degener and this is bioelectronics. In this lecture I want to cover optogenetics and what this means in terms of cellular vision. So obviously I need to describe the concept and then I'll describe the molecular basis for optogenetics, and then I'll discuss the three types of channels or GPCRs and pumps that form this structure molecularly. So, first of all, the concept. So far in the course, we've talked about nerve cells, and we've talked about how nerve cells receive chemical and electrical inputs, and those inputs cause the cell to integrate the signals, and then out pops a signal somewhere else. I've also mentioned that there's other types of nerve cells which have sensory functions, for the eye and for pressure in particular. The concept of optogenetics is that you take a gene, you insert it in some kind of vector, a method for delivery, and you insert this into the brain and you make cells, nerve cells, which aren't meant to be light sensitive. You engender light sensitivity in those cells so that they can be activated by light. And conceptually what this allows you to do is to say, well, okay, I want blue light to activate a certain cell type, or red light to activate another cell type, or perhaps to use blue and red to activate just one cell type, but one that excites it and one that inhibits it. And conceptually, you can see it in this particular video. This is a video of from some years ago. This is of a mouse, and if we pause it for a second, this mouse has an optic fiber coming from external to the head, and the optic fiber is illuminating a part of its brain which decides motion, and this particular part of its brain has been optogenetically photosensitized, so those neurons will be activated by light. And then when the light turns on, you can see a tiny little spot on the brain there. When the light turns on, the rodent is running, it's activated, and then when the light turns off, the rodent is no longer moving. So that's a very, very simple concept of this technique, which is very powerful. Now, in order to understand, well, how do we bring light sensitivity to nerve cells, let's review again the operation of neurons. So this is a myelinated neuron. It has dendrites which receive inputs. Those inputs are all integrated at the axon hillock, which decides an action potential, and for a myelinated neuron, the action potential jumps from node of Ranvier to node of Ranvier, until eventually it comes down to the bottom of the axon, where it then synapses with whatever the target may be. Now, the basic achievement for these kinds of activities are ion channels, and ion channels are electrically gated channels, or we can have receptors, which again are basically the same thing. They are ion channels, but they are chemically gated. We can have pumps, which are there to pump out sodium, pump in potassium, and basically regulate the polarization state of the cell. And then we've mentioned these metatropic type receptors, or GPCRs, which are are particular sort of molecular proteins on the surface of the cell which accept a chemical, which then in turn activates a G protein cascade, which then either activates or deactivates multiple ion channels. So these are the possibilities we have to potentially photosensitize our cells. And naturally, I mentioned before that GPCRs are the basis of vision in animals. So let's have a look. We do have cells which are sensitive to light. They sit in our eyes. And if we look at the two types of vision, in the animal kingdom, we have vertebrate vision and we have invertebrate vision. And molecularly, they're quite different. It's not just that the structure of the eyes are different, but actually the molecular process is quite different. For basically all mammals, we use a protein, that GPCR protein, called rhodopsins. For invertebrates, they use a protein called metarhodopsins. And it's a bit like Max. So, the old Apple Motorola chips versus the Intel chips. They're kind of similar, but they're the opposite way around. But also in the animal kingdom, you also have vision. So there are algae, these single cell algae, that have something called channel rhodopsins. And these are literally individual molecular eyes on a single-celled organism. And it's also worth pointing out that bacteria can also have, in a sense, vision. You could argue it's more like a solar cell than it is vision, but they have molecules which are activated by light, which are there they use the energy from the sun to basically perform a particular task, a bit like a solar cell, as opposed to a sensing function. At the core of all of these, whether it's plant-based, whether it's mammalian-based, at the core of all these molecules, rhodopsins, whether they're channel rhodopsins, metarhodopsins, or whatever, is vitamin A. And vitamin A is basically this molecule. It's a conjugated small molecule, and it looks like a benzene ring with a tail. And when it's activated by light, now this is a very simplistic form, it actually has multiple different states and multiple different activated states and so forth, so chemists might not particularly like this description, but when it's activated by light, effectively the tail is wagged like a happy dog, and this wagging of the tail, this activated state, can then activate the overall rhodopsin molecule, whether that's channel or meta or whatever, and then that overall activated molecule can then perform the function that we'll describe in a moment. Now, in the case of mammalian vision, we have what are called rhodopsins. And what rhodopsins do, this is a bit like the description I mentioned before with GPCRs, except now that light comes in, and the light comes in and activates the rhodopsin cell, the rhodopsin protein, and the rhodopsin protein then activates this G protein component, GDP. That in turn starts a chain reaction, and to cut a long story short, what it does is it converts cGMP back into GMP, and cGMP is used to hold ion channels open. So when you remove the cGMP, some of these ion channels close, and then the pumps start pumping out the sodiums, if you like, and the cell hyperpolarizes. Now this is a really, really weird structure, because in essence, it's a bit like having a house where you have the heating on full blast and the doors open, and in order to then heat your house, you close the doors, and then when it gets too hot, you open the doors again. It's a very strange concept from an energetic perspective, but it does seem to work for us mammals. Invertebrates have a somewhat more sensible approach, and in their case, what happens is it's kind of the opposite. They have another G protein type cascade, but the cascade results. So if this is your if this is your activated rhodopsin protein, so you've got your rhodopsin, metarhodopsin, it causes the opening of what are called TRP channels. It's a particular type of channel. So this is a more sensible approach, that you have your standard cell, and you get as the channels open up, it gets depolarized, just as I've described before with neuron cells. Whereas in the case of the human eye, we have the opposite. We have that concept of where you've got a heated house and you leave the doors open. Very, very strange, but this is how we've been developed. Now, if we want to think about what this means from a mathematical perspective, we've already described the metatropic receptor, and this is basically the same in that you've got your standard conductance for an individual ion channel. It's the conductance times the voltage difference of the voltage across the cell and the reversal potential, and then you've got an integral function, and then you've got the number of cells being activated. And in the meta-rhodopsin case, it's basically the same. The difference between them is that in the case of rhodopsins, what we're adding up is the not transported ions, and in essence then the cell can be hyperpolarized. And in the meta-rhodopsin case, it is just the transported ions which help depolarize the cell. If we move to plant type channels, then in effect you get something which looks much more simple than the G protein cascade that I've described before. You end up with these very, very simple channels, channel rhodopsins, where effectively the opsin is connected or integrated into the channel, and then when light comes in, the channel opens up, and then you get your standard ions flowing through as before. So the kind of conceptual equation of transported ions is very much that Hodgkin-Huxley type relation, the conductance of the channel, the voltage difference, and the time integral. And these exist in these small little algae called Chlamydomonas reinhardti. And this was the discovery of this particular channel was the basis for what is the optogenetic revolution. But you also have what I mentioned before by bacteria and pumps. So you've got these are bacteria, these purple bacteria or red bacteria. They can exist in salty marshes and lakes, and because they live in either salty or perhaps acidic environments, they have to get rid of ions. Now, in some cases it's hydrogen ions, in some cases it's the sodium ions, and in order to do that, especially for the chloride ions, they use pumps. Yeah, and basically what happens in the dark, the pumps do nothing, but in the light, the pumps pump out the chlorine from the cell. Very, very simple. And if we want to think of our model, well, one absorbed photon leads to one transported ion. So if we were to summarize these different approaches, this would be the least efficient. You'd get the fewest number of ions per photon, whereas the rhodopsins would give you the most amount of ions per photon. Okay, that's it for this lecture. In the next lecture, I will talk about this in more detail. Thank you very much, and see you at the next lecture.